So during metaphase 1 of meiosis 1, side note, notice so far it's been prophase 1, prometa 1, meta 1, spoiler alert, anaphase 1. Later in this chapter where it's going to be prophase 2, metaphase 2, anaphase 2. The 1, the 2 is to denote which half of meiosis you're discussing. So metaphase 1 is the first half of meiosis, which means we're looking at separation of homologous chromosomes, the homologous pairs. So the homologous chromosomes have been lined up 4 high, the tetrad, and during metaphase 1, they're going to be arranged, near the equator. Okay, it's still going to see the same thing we talked about with mitosis. The centrosomes, the mitotics, the spindle formation, the attachment to a microtubule spindle at the kinetochores. Same thing with the proteins. Can I think of what proteins were? The ones that walk, motor proteins. Same thing, motor proteins and a protease chopping up the microtubule, all of that. Same thing. All of that works the same here. Now, here's the big push for genetic uniqueness. So far, we have talked about the possibility of some recombination occurring due to that chiasmata, the crossing over. We also see it right here. Look at this. Notice just with 2 chromosomes showing, we see a big difference because how they line up as the tetrad. Notice at the top example up there. All the blues on one side, all the reds on the other. Down at the bottom, this example, blue, red, red, blue. Now, carry out this, which way they line up, through all 23. So you get 2 to the 23rd, I think it's like 2 to the 23rd different variations of how this could occur. That's a lot. Because then you go through meiosis 1, meiosis 2. You end up with haploid cells. Here they're all blue. Here they're all red. And down here, a mix of blues and reds. Anaphase 1, telophase 1, cytokinesis. Same thing we talked about with mitosis, okay? The pulling of the part, motor proteins pulling, walking up the microtubule, anything attached to it gets pulled along with it. As they step along, the motor proteins step along, protease chops it up, blah, blah, blah. Telophase 1, as soon as the sister chromatid pairs are pulled far enough apart, or the homologous pairs are pulled far enough apart, the sister chromatids, all 46 of them, will then be, wrapped up again in a nuclear envelope. And then you get cell division, the cytokinesis, just like we talked about before. Video, click on the link, upload, take a look at, show this. You also y'all can go back to, wow, take the boy out of West Virginia, can't take the West Virginia out of the boy. If you go and you look at the web page in Canvas for this chapter, you'll see that I have also embedded some YouTube videos that I think really show